Hello, students. Hello. Now, again, I'm with you. Two or three students have already joined me. Good, very good. Means Naveen, and after that, Harsh is waiting for me. Good, very good. Where are rest others? Pratyaksh, Hanshika, good. Good afternoon, Vida. Good afternoon. Okay, one more connected. Who is this? Ayushi. Good. Good afternoon, Vida. Okay, Harsh, Janvi, good, very good. Yes, Prakash Singh, yes, Prakash Singh. Welcome, beta, welcome. Yes, I know, beta, I know. Rishabh, good, good, beta. Good afternoon, good afternoon. Once you join, don't leave the class, okay? Just continue till the end. Naveen, okay, good afternoon, beta. Priyam also, good afternoon. Tenar Actu now. Harsh Pandey, okay. Kaushal, experiment beta and change your ID name. Okay, this will help me to recognize you, and also it will help me to fill your attendance. Yes, someone of you have left the class. No, that is a bad thing, very bad thing. Once if you have joined the class and don't leave the class before end. Priyam, hello sir, hello beta, hello. Yes, let everyone to be joined, and I'm going to maintain the board. Yes, sir, my name is Kaushal. Okay, you are Kaushal. Okay, from which class, Rida? Yes, Kurkat, you are also here. Good. Harsh Pandey. हाँ बेटा डेली जल्दी क्लास शुरू होगी. Your class will begin at sharp 12:15. Way. Your class timing is 12:15 to 1 o'clock. Yes. 12-hour online at present. Think there are 150 students, including all the three section. Okay, out of which only 12, 15 are coming online. I'm also going to inform regarding this to your school campus. Okay, they will contact you personally. Don't worry. Those who are not attending the class, against them, principal as well as school administration will take a strict action. That's why NC Yadav. Who is NC Yadav? Good afternoon, beta. Kaushal, you are from 9B1. Good. Okay. Okay, beta. As in previous class, we have discussed about the discovery of cell. Okay. And also we have discussed the cell theory as well in new cell theory, everyone. Some of the students have doubt in new cell theory. What is difference between cell theory and new cell theory? Okay. So I'm going to repeat this topic again. Uh, some also so many students of you have sent me the homework in a WhatsApp group. I have seen and get means feel too much happy that you are responding to me. Uh, that is good thing. Okay, and now keep it up. I just want to say keep it up. There are so many girls. I want to girls and boys both who have done homework. I want to take their name. Shreya boys. Praveen Singh, okay. Praveen Singh says Arun Rai. Arun Rai, who's okay? Arun Rai is Smriti. Smriti from ninth A. Good. In ninth A, even boys. Vikas Tripathi. Have done. Ninth, even boys are not doing their work. Okay. None one of you have done the homework. Means okay. Uttam Pandey. Good afternoon, Buddha. Good afternoon. Vikas Tripathi. Hi, sir. Hi, Buddha. Naveen Singh. Naveen Singh, I am here. Okay, good, 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 Naveen. Continue class. Yes, good. Okay, that's why everyone do your work and send your means, pick of your done homework in group, in your respective group. Okay. Yes. Yes. Now I'm going to start the class. Yes. Okay. Hmm. Then in previous class, I have explained the cell theory as well as modern cell theory. In cell theory, it was given by Sleden and Schwann. Sleden was a German botanist. He was a botanist, and after that, Schwann was a geologist. Both has worked separately, and there was a gap between 1 year in both works. But they had compiled their work and formulated a cell theory. This cell theory states that all the living organism means plant animals are made up of cell, and this cell is the basic structural and functional unit of life. Okay, and after fewer virtue has discovered one more thing, that all living cell arises from pre-existing cell. That's why now he added this statement also in cell theory and formulated new cell theory. New cell theory have three statements. One is all living organisms are made up of cell, and cell is the basic structural and functional unit of all living organisms. Okay. In this way, the third statement was statement is all living cell arises from pre-existing cell. Yes. Okay, so many, and now I'm going to just solve the homework question I have given. The first homework question was what is cell biology? Okay, the cell biology is the branch of biology today. Okay, now another question was what is protoplasm? The living substance present inside the cell is termed as protoplasm. This protoplasm term was coined by Purkinje. Okay, and protoplasm is only the thing which results as a life in a cell. A cell will be dead. It will be non-living thing if it lack it will lack the protoplasm. Okay. Another question was what is cell theory? As I have told, there what is cell theory? Yes. What is new cell theory? What is difference between cell theory and new cell theory? Everything I have explained. Okay. Now I'm going to start the next topic for today. That is instrument for studying the cell. Studying the cell. Okay. Which kind of instruments are used to study the cell? Okay. Can we see the structure of cell with any instrument? Or any specific instrument is required to see the structure of cell? Yes, we can't see the structure of cell with the help of any instrument. We need some specific kind of instrument devices to study the structure of cell. Okay, so now we will discuss which kind of means instruments are widely used to study the structure of cell. There are two kinds of devices. Number one is a light microscope. Okay, and number two is electron microscope. Okay, this light microscope is a light based microscope. Microscope means micro. Here, first understand the meaning of microscope. Okay, microscope scope means a device to see a thing. Okay, like telescope and a bioscope. So many. It is a device which is used to see different object. And micro means means scope which is used to see microorganism. Means the thing which are present, which are in micron. Which dimension is in micron? Micron is a unit of length. Micron is a unit of length, that is micrometer, and 1 micrometer is equal to 10 power minus 6 meter. It means 0.0000001 meter. Okay, 1 micrometer is equal to 10 power minus 6 meter. It means it is too much smaller unit. Okay, and to see this comes object of this dimension which have okay उल्टा दिख रहा है. 1 minute, I'm going to correct it, don't worry. Now it's I think this will be clear to you. You should tell me yes, now I had corrected. I had corrected. Okay, you should inform me earlier, na? Now it is clear for you? Yes, good. Good. Okay, there are two kinds of means where to study the structure of cell. We uses a microscope. Microscope is a device which is used to see the things which have a dimension of 1 micrometer. 1 micrometer. And 1 micrometer means micrometer is the length which is equal to 10 power minus 6 meter, that is 0.0000001 meter. Okay, it is too much smaller unit, and the things which have a dimension of 1 micrometer are too much smaller, and they can't be seen with naked eye. Okay, to see this structure we need a special kind of device that is termed as microscope. Okay, there are two kinds of microscope which are being widely used nowadays. This is a light microscope, and another is electron microscope. Okay, light microscope is based on is an optical device. It is an optical device, means it works in the presence of light. Okay, while electron microscope is a electricity based device, and it works in the presence of only electricity. Without electricity we can't operate this device. Okay, now light microscope, there are two kinds of light microscope also. One is a simple light microscope, and another is compound light microscope. Compound light microscope. Okay, both runs with the help of both are based on both are optic device, and they runs in the presence of light. Okay, then is light. Okay, the simple microscope, the example of simple microscope, we can say that a hand magnifier. Hand magnifier is a simple light microscope, we can say. And after that, compound microscope is available. I think it will be available in your school laboratory. Okay, Naveen asking, sir, what is light microscope? Repeat it again. Light microscope means the kind of microscope which works in the presence of light. Okay, they are an optical device. Yashashwa group, good afternoon. Good afternoon, beta. So many students are joining the class, and as well as they are leaving the class. But this is bad thing. Okay, once you have joined a class, then continue till the end. Now it is clear what is Naveen Singh? Yes. Naveen Singh question, what is a light microscope? Then light microscope is a type of microscope which is an optical device, means it works in the presence of light. Anything is visible under this microscope, only light is present. When only light is present. If light is not present at there, then we can't see anything with the help of this light microscope. This light microscope are two types. One is simple light microscope, and another is compound light microscope. Okay, the simple light microscope, the example of simple light microscope, maybe we can say it is a hand magnifier. Hand magnifier. Okay, hand magnifier. In this way, compound microscope is available in your book, and it is also given. Its a diagram is also given in your side book. I have forwarded in the WhatsApp group. Okay, you may visit to the book available in the WhatsApp group, and you can see at there what is light microscope and compound micro. Both diagram is visible at there. Okay, another kind of which is most important microscope. And one more thing, whatever the discovery we have means discussed in previous class, all was made with this simple microscope. Okay, till now we have not discussed any discovery by the compound microscope, or that is electron microscope. Okay, all the discoveries which was made in earlier days was done with the help of simple microscope. Some of them are also done with the help of this compound microscope. Okay, so now another this magnification. Now we should discuss what is the magnification means. What is the magnification power of these things? Okay, so this means Arun Rai is asking page number. Page number, beta. Page number, you can just turn the page, you will find it. The heading is given. For this, I have to search this book. 1 minute. I will also tell you on which page. Wait. I'm also giving the page number to you. Priyanshu Maurya. Okay, so Priyanshu, you are also here. Subhash Maurya saying good morning, good afternoon, beta. Day by day your number is going to. One more thing for all students, subscribe the channel and press the bell icon. Okay, subscribe the channel and press bell icon. This will help you, as well as to me for notifying whenever I'm going to be live again. Okay, simple and compound light microscope. What is difference between simple and light microscope? Okay, Naveen Singh is asking. Yes, 1 minute, beta. Page number 133, 30. Yes. Yes. The figure of a light means simple microscope is available in ebook on uh, page number 212. Yes, page number 212. Open the PDF file and see at page number 212. At here it is means figure of light, simple light microscope is given, which was made by Anton van Leeuwenhoek. Okay, and after that the figure of compound microscope is available on page number 215. Mark, and this is available on page number 213, and this is available on page number 215. Yes, page number 213 simple light microscope, and on page number 215 compound light microscope. You can see it there. So page number 200, but in PDF it is showing 213. Okay, no problem. You can see it there. Yes. Now, So, what is difference between simple and light microscope? It is there is two difference. First thing, the magnification power of a simple microscope is very less. It can magnify about 10 to 100 times, while this compound light microscope can magnify about 1000 to 1500 times. Okay, both are light based microscope. Arun Rai is saying I NCRT book. No, beta. See in side book I have forwarded in the group. Okay, Arun, are you in group? Have you joined the group or not? Arun Rai. Okay. So now another kind of microscope which is used in the field of cell biology. It is electron microscope, and this microscope is Chandan Upadhyay is saying says sound is not clear, voice is not clear. Others, for others, my voice is not clear. Reply me, hurry up. Lavin is saying it's okay. What? My voice? Good. I think Chandan, then you have some network issue. Okay, that's why your video may buffer. Okay. Okay, Chandan, correct your network, means solve a network issue. I think you have network issue, that's why. Okay, now the electron microscope is another kind of microscope which runs on the basis of with the help of electricity, and its magnification power is 1 lakh to 5 lakh. 1 lakh to 5 lakhs times. Okay, it means my electron microscope can magnify anything about 1 lakhs time. Means a very fine object, means we can say if any object have a dimension of about the point of a needle, then it will look like a Mount Everest in under a electron microscope. That's why it will be too much big. Okay, 1 lakh time means 1 lakh time, 1 lakh to 5 lakh times dimension can be increased by with the help of electron microscope. Okay, so this electron microscope helps in the study of ultrastructure of cell. Ultrastructure. Okay, ultrastructure of a cell is studied with the help of this electron microscope, while light microscope is used to study the structure of cell. Means we can't study what are the different other structures present in class 7th, means class 8th you have studied inside a cell. Inside a cell there is a nucleus, and after that cytoplasm is found in this cytoplasm there are so many other structures are present. They are termed as cell organelles. You have studied right there. These cell organelles, structure of these cell organelles can't be studied with the help of this light microscope. To study the structure of this cell organelles we need electron microscope. Okay, after the discovery of electron. Okay, in Knoll and Ruska, there was two scientists. It was two scientists. One was Knoll and another was Ruska. in 1940, they have discovered the electron microscope, and this electron microscope is now used to study the means structure and structure of other cell organelles present inside the cell. Okay, these are the two basic, and after that we also use uses as some other devices to study the structure of cell under a microscope. This is a glass slide. Okay, this is I'm going to explain other things. Like one is a glass slide. It is a rectangular glass piece. Okay, with it we need a cover slip. It is a glass slide, and there is a cover slip which is either in a circular form or a square lid. Square lid. Okay. With it, we also need a petri dish. And watch glass. This is watch glass. These are cover slip. Okay. This is petri dish. This is watch glass. Okay, with it we need forceps, needle. Okay, forceps I have seen chimti, it is chimti, and after that needle, brush, and some pigments, some means stains are required here, like saffronin. Okay, and after that methylene blue. Okay, these are the some pigments, I mean stain. These are the stains which are used to mount the specimen. Okay, specimen is the thing which we are going to means which will be seen under the microscope. Okay, this means cell is too much fine object that it appears transparent. We can't see it even under light microscope or compound. That's why to see it first we have to कलर इट, हमें इसको कलर करना पड़ता है, इसको रंगना पड़ता है। रंगने के लिए हमें कुछ स्पेशल काइंड ऑफ कलर हमको चाहिए होते हैं, जिनमें एक सैफरन होता है, दूसरा मिथाइलिन ब्लू होता है। दे आर वाइडली यूज्ड इन लैबोरेटरी टू stain the specimen. Okay, so these are the different things which are used in laboratory to study the structure of cell. Okay, yes, for now in a classroom when I will attend your means laboratory, then I will explain how the slide of onion peel. Someone was asking about the onion peel in group yesterday. Oh, what is onion, how we prepare the glass slide of onion peel, and how we observe it under the microscope. In this way, how we will also discuss at that how we will prepare the glass slide of chick cell. Okay, chick cell means a cell from this side. We just take cell from this side, and after that prepare its slide and observe its structure under the microscope. Okay, who is not active idea, who's is Adil Alim is here. Is Adil Alim is here from class 9th B1. Adil Aim, are you watching me? Can you hear me? If you can hear me, then reply here. Okay, these are some other devices which we use in the study of cell under microscope. Different kind of microscope, like simple light microscope, compound light microscope, or electron microscope. Electron microscope. Okay, electron microscope was invented by Knoll and Ruska in 1940. Okay, so before 1940 we haven't seen what which kind of structures are present inside the cell. These means this is the structure of cell. Means if it is cell, this is its a nucleus, and around the nucleus so many other fluid-like structure was seen by the Anthony von Leeuwenhoek and also by the Robert Brown. Okay, but which kind of structure are present in this was not seen by anybody before 1940. Okay, after the discovery of electron microscope, that is in 1940 by Knoll and Ruska, we become able to study the different organelles present at here. Okay, other structures present at here. Okay, it's a clear. Not till everything is clear. I think it should be clear to everybody. Today 24 students or 25 students are attending the class. It is very good thing. Just increase the number. Day by day increase the number. I want to see here about more than 100 students daily. Okay. And every day I want to see here 150 students daily here. Inform everyone. Call to your classmate and say them to attend the class. Now this number become 26. I am continuously watching the number. Don't worry. I will note down your name well and will send this information to your school principal. Don't worry. Day by day reporting will be given at the. So soon you will receive call and after that messages from school. Those who are not attending the class. Okay, now I think everything is clear. Now I'm going to move ahead. Now I'm going to just cover next topic. Yes. Another topic I'm going to take here, that is type of cell, type of organism. Organisms on the basis of number of cell in their body, okay, how many kinds of organisms are found on the basis of number of cells in their body. Okay, so there are प्रियम आस्किंग सर, न्यूक्लियस के अराउंड में जो है उसको क्या बोलते हैं? उसको बेटा साइटोप्लाज्म बोलते हैं. क्या बोलते हैं? साइटोप्लाज्म. Okay, and cytoplasm as well as this nucleus are collectively termed as protoplasm. Okay, protoplasm means this cytoplasm and nucleus, cell membrane, all are the living substance of the cell. Okay, they are living thing, while cell wall, which was observed by Robert Hooke, was non-living. Okay, now so now we'll discuss type of organism on the basis of number of cell in their body. So on the basis of number of cells in organism's body, there are two kinds of organism. First is unicellular and another is multicellular. Unicellular and multicellular. Okay. Now I will explain one by one what is unicellular and what is multicellular. Okay. The unicellular organisms are those organisms which body is consist of only one cell. Body is consist of only one cell. Okay. For example, we'll take some example like paramecium, euglena, plasmodium, okay, yeast, okay, rhizobium. I think you have heard the name of rhizobium. How many of you know have heard the name of rhizobium? How many of you have heard the name of rhizobium? Okay, Turkesh is saying me, sir. Good, good, beta. Anyone other? Me too. Okay. Vishakha Singh saying me too, sir. Good, very good. What is rhizobium? What is rhizobium? Ansika is saying I have heard about it. Good, good, beta. Utsav Singh has WhatsApp me. He is not attending the class due to network problem. But correct your network issue. Just solve this network issue. Otherwise, it will be very bad for you. Okay. Okay. Rhizobium is a nitrogen fixating bacteria. It is a nitrogen fixating bacteria which fixes the atmospheric nitrogen into the soil, and it awaits the atmospheric nitrogen for plant to synthesize protein, okay, amino acid. Okay, this is one kind of organism. Another is a multicellular organism. Okay, multicellular organism means the organism which body is consist of more than one cell, more than one cell, one cell. Okay, like all higher plants and animals, all higher plants and animals are multicellular or multicellular. Okay, so now think what is a unicellular, what is a multicellular, it is clear for you. Okay, so your time is up now. I'm going to end the class now, but again I will meet you at same time, that is at 12:15 p.m. Okay, and one more thing I'm again going to repeat. Subscribe the channel and press the red bell icon. Okay, this will help me as well as to you for to notify whenever I'm going to be live again. Yes. Okay. Goodbye and good day. Okay. And one more thing, stay safe and stay home and enjoy your life. Good day. Good day.